So there are 3 lines of defense when it comes to our immune system, being able to recognize things, being able to fight things off. And, these is all things to try to keep you healthy. Of the first 3 lines of defense, the very first line of defense is just the barrier. Let's just prevent things from getting into the body. If they can't get in, they're not going to cause issues. And so our first line of defense are all the external physical barriers, your skin, and all the different ways our skin helps us keep helps us keep everything out, as well as our mucous membranes and our digestive tract, our respiratory tract, all ways just try to prevent things from getting in. The second line of defense are internal processes that also try to keep us healthy. There's different types of white blood cells that try to keep us healthy. There's different chemicals that our body makes that try to keep us healthy. There's processes like phagocytosis that try to get rid of pathogens that may have gotten in. What the first and second line of defense have in common is they are what's known as innate defenses. Innate just means things we were born knowing how to do. It's like you were born with skin. Yep, it knew what to do. You were born with certain white blood cells. You were born with cells knowing how to do phagocytosis. They didn't have to learn how to do it. They didn't have to teach or anything else. So your first 2 lines of defense were just all things that our body was born knowing how to do. That's, if you haven't watched that video and taken the quiz, is all about those. Today, we're going to get into the adaptive immune system. So this is where it recognizes very specific invaders that may get into the body. It will activate certain cells. It will target those invaders. It will destroy them. And then the benefit of it, it will then form some memory cells. So if you ever get exposed to things again, your immune system knows how to get rid of them and hopefully get rid of them faster than they can reproduce. And ultimately, you don't get sick from things over and over and over again. The white blood cells that we're going to be talking about in our adaptive immune system are the white blood cells called lymphocytes. All of the other white blood cells are all part of your second line of defense, but the lymphocytes are the ones that are very specific and can form memory. So just breaking it down into kind of those 3 lines of defense again, our innate defenses are those first and second line of defense. That very first pink bubble on the top are all our surface, prevention of getting things in. The second line of defense are all the internal defenses we're born knowing how to do. Those phagocytes, things called natural killer cells, inflammation, other types of proteins, a fever. That's something our body was born knowing how to do. It's part of our immune system response. But it's the adaptive immune system, the third line of defense that we're going to talk about today. Now, the adaptive immune system works in 2 different ways, depending on what kind of invader got in the body. There's a whole branch of how it works called your humoral immunity, which is my plan to get through all of today. And then we have a whole branch called cellular immunity, which is my plan to get through on Thursday. So just notes, heads up. So in the PowerPoint between today and Thursday, we're talking a little bit about what is adaptive immunity, where does it take place in the body, what are the different players in the game, what are B cells, what are T cells, what are antibodies, what are antigens, what are cytokines, what are something called MHC, which we'll get to today, what's something called APC or antigen presenting cells we'll get to today. And then we break it down really into those 2 big processes and how those players all work, the cell-mediated and the humoral. I really should flip it because I'm going to talk about the humoral first. And then based on all of it, how we get that acquired immunity. It's kind of the reason why we get vaccines. So we'll talk about those too. So first, just what is the adaptive immune system, or what is adaptive immunity? Well, it's the body's ability to recognize and defend itself against distinct invaders, meaning very specific things that get in the body, and possibly any of their products, certain invaders, whether it be a fungus or a parasite or any kind of bacteria. Some produce toxins. So not only can we recognize the bacteria, we could also recognize the toxin that they make. So it's not just recognizing the pathogen. There are 5 attributes. Let me put them over here. Of our adaptive immune system. I'm going to try to make it brief as possibility. The first one, the specificity, is our adaptive immune system can recognize specific invaders. And maybe not even just recognize and kill. Like any random thing that can get in the body, our body can recognize it. Like as a like a piece of dust somehow gets in the body, it knows exactly what that dust particle is. Inducibility. Our adaptive immune system is patrolling the body, but it's not actively fighting anything until you get something in the body. The inducibility is that the specific invader, you could even draw an arrow if you want. Induces or activates cells of the immune system. So we have white blood cells that are patrolling. Our lymphocytes are patrolling the body, constantly looking for any possible random invader that may get in the body. It's not actively doing anything, it's just patrolling. It's just looking. But once it recognizes that specific invader, then it becomes activated. Once activated, or induced, makes clones of itself. If you've got a white blood cell in your body that's patrolling, since we're going to test for strep bacteria today, if you've got a white blood cell in your body that is patrolling for Streptococcus pyogenes, the bacteria that causes strep throat, it's just patrolling. But if you would have that bacteria in your body and it recognizes it, it's like, oh, that thing actually got in. Once it recognizes and it attaches to it, it becomes activated, and then it starts to make more copies of itself. It's like, I'm the cell that needs to recognize it and fight it off and kill it, and the best way you win a war is if you have a whole huge army. And so it makes a whole bunch of clones of itself that can recognize all of those strep bacteria that got in and target them all for destruction. Number 4, unresponsiveness to self. Ignores normal body cells. Our immune system has to be able to recognize foreign invaders and ignore everything that's normal. If it doesn't, this is when we get into some of our immune system disorders. That there are times, sometimes the immune system will recognize normal body cells as foreign objects and target them. And then we end up with unnecessary inflammation and other immune system responses. And then 5, we have memory cells. This allows us to, activate quicker with each exposure. Meaning any other time that you get that same pathogen in your body, your immune system recognizes it faster and faster and faster and can target it for destruction, hopefully before it ever takes hold, before it ever causes any issues whatsoever. Is it always perfect? No. And there are certain organisms, certain microbes that trigger more memory cells versus other ones. If you've had strep throat before, can you get strep throat again? Yeah, the memory cells do not last very long for certain bacteria, whereas other ones, they do last a really long time, which is why if you've got some vaccines, you're pretty much protected almost your entire life from one vaccine. Yeah, some of the main players are our lymphocytes, because I've already been talking about them. It's of all of our white blood cells. It is one kind of white blood cell, the lymphocytes, but there are 2 types of lymphocytes. Underneath a microscope, they would look identical. You would have no idea which type of lymphocyte it is. You would just be like, oh, it's a lymphocyte. Underneath the microscope, a lymphocyte has a massive nucleus. So this dark purple inside the lighter purple, this is the nucleus. Like it almost crowds out the entire cell. And it's very grainy. That's just how you identify a lymphocyte. But there are two types of lymphocytes. There are B lymphocytes and T lymphocytes. We just shorten it to B cells and T cells. It's just easier to say. Why they call them B and why they call them T, it's based on where that white blood cell matures. So where does that white blood cell, where does that lymphocyte go to mature? If it's a B cell or B lymphocyte, it matures in the bone marrow, hence it's B. If it's a T cell or T lymphocyte, it matures in the thymus, hence the T. So it's not like it looks like anything, it's just where they mature. Now I'm going to add on to here. I'm going to erase this first. Whiteboard's not super tall like the lab. So our adaptive immunity I mentioned a little bit earlier in the PowerPoint is there's two branches. There's a humoral branch or system on how it works, and there's a cell-mediated branch on how it works. The humoral branch uses B cells. And this is the branch that we're going to look at today and talk about how B cells actually work. The cell-mediated branch, which we go over on Thursday, all has to do with T cells. So the humoral branch and our B cells, that's what we're going to focus on today. One of their main goals is making antibodies. T cells do not do that. I'm going to add a little bit more up there on the board. So the cell-mediated branch, they use different kinds of T cells or T lymphocytes for recognizing foreign invaders. And they recognize different kinds of foreign invaders, and they kill them differently as well. More about both branches as we get into them. It's the immune system and all these lymphocytes. These T cells and B cells are all part of the lymphatic system. Your lymphatic system is your immune system. I was going to say, there is a lymph fluid that you have lymphocytes that are constantly floating around your body and patrolling. So your lymphatic system is your immune system, which confuses a lot of students. They're like, what is a lymphatic system? What's the immune system? What's the difference? There's not. It's a lymphatic system, which is the immune system. Now, the lymphatic system, what it consists of, you have all the lymphatic vessels and lymph that's floating around in all of those lymphatic vessels. Lymph has a similar composition to blood plasma. I'm actually going to go back a second. And if you notice all these green lines streaking all over the body, these are lymphatic vessels. And lymphatic vessels pretty much mirror wherever a blood vessel is. If there's a blood vessel going somewhere, there's a lymphatic vessel right next to it. And so the fact that it has a composition similar to blood plasma is because when you get overhydrated, a lot of that fluid from your blood will just go right into the lymphatic system. But it also works great because they run side by side, is that if something would be in your blood, which is never good to get anything in your blood that's not supposed to be there, you have lymphatic vessels that are right there that are also patrolling your blood supply. So the lymph will travel all over the entire body, and the job is to patrol it. It is looking for foreign invaders that get into different areas of the body. Now, other than just having all the vessels everywhere, there are a couple lymphoid organs, lymph nodes, different areas where you usually have really high concentrations of blood cells, specifically the lymphocytes. And so we have what are called primary lymphoid organs and secondary. The primary is just where those lymphocytes matured. Again, bone marrow for the B cells and the thymus for the T cells, where we get our B and T. The secondary lymphoid organs is where we have a really high concentrations of white blood cells. And so if one of these lymphocytes recognizes something foreign, a lot of those white blood cells are hanging out on the lymph nodes and attacking and getting activated. This is one of the reasons when you're sick, you have a big old lymph node right in your neck, and you're like, oh, I feel like I'm getting something. I can feel my lymph nodes are swelling. Well, yeah, because you've got lots of white blood cells that are all attacking whatever foreign is in your body, and a lot of that is going right to those lymph nodes for recognizing, activated, and killing that pathogen. Your lymph nodes, which we've got multiple ones all over the body, big ones right in the throat that you can feel quite often. We've got a couple big ones in the groin, in the armpit region, our spleen, our tonsils, and then something also called our mucosa-associated lymphoid tissue, which is a mouthful to say. The big part is, so it's mucosa-associated. So pretty much this is going to be found, it's just a tissue found in locations where we've got mucous membranes. And if you remember, mucous membranes are the membranes that are exposed to the outside. So your digestive tract is exposed to the outside because whatever you put in your body, the outside, goes right through the digestive tract. Your respiratory system, you're constantly breathing things in, and that mucosa membrane, part of our first line of defense, really has to be able to recognize foreign invaders. Because your respiratory system is exposed to outside stuff, because your digestive tract is exposed to lots of outside stuff, we want a lot of white blood cells right there. We want those white blood cells right there, recognizing anything that might get in the body. Is there a lot of bacteria in your digestive tract? Again, it's like, E. coli. There's so much bacteria that's in your digestive tract. Those bacteria can be doing wonderful things as long as they stay in your digestive tract. If they leave your digestive tract, if they can get past the mucous membrane, then they're going to cause a lot of issues. And so our body is equipped to have lots of white blood cells right there at the digestive tract, looking for anything that's trying to get past the digestive tract. Now, another term that we're going to talk about a lot are antigens. An antigen is a molecule that the body recognizes as foreign, generally. Now, how the body recognizes something as foreign is that on the surface of that object, on the surface of that antigen, that foreign thing, there are 3D structures called epitopes. So an epitope is just a 3D structure on the outside of an antigen. Use this little bit of whiteboard space down here. I use a big blue blob, but I don't have blue. But if this big, huge structure is the antigen, that is the foreign object, the body was like, okay, yeah, that's foreign. But I only know it's foreign because on the outside of that object, there are little 3D structures sticking up that the body was like, I don't know what a little red triangle is. That's not supposed to be there. It's the body recognizes that as foreign. Those little orange circles aren't supposed to be there. Those little green rectangles or squares aren't supposed to be there. Those are little 3D structures that the body recognizes as foreign objects. Because when the body recognizes something as foreign, that means when it recognizes it, it can attach to it and target it for destruction. This is just showing different pictures, different examples. Again, if you've got a foreign object, you can have one small area, even on the cell. Again, if a bacteria gets in the body, maybe it's not the entire bacteria that our immune system recognizes. There's some structure on the bacteria with little tiny shapes on the outside that's like, oh, I know what those things are. Those are that one bacteria that got in the body that one time, and we know what to do about it. Watch the little video. Just shows a picture of an antigen. But there are different types of antigens that our body can recognize. Again, foreign objects. So there are what are known as exogenous antigens Exo means outside, like an exit, you go outside. These are outside antigens. So these are structures on the outside of things that our body can recognize as foreign. So there's little structures all over these little outside structures on the outside of structures. These are little structures that, yes, are on the outside, easily presented. Our immune system can see little green triangles because we don't have a flagella that looks anything like a bacteria. There are structures on the flagella. There are little shapes on a flagella. Body can recognize it as foreign. They can recognize these little blobs or these little blue circles all as foreign objects. An endogenous antigen means this is an inside structure that has to get presented on the outside. This becomes an issue with viruses. If a virus, when it's reproducing, is inside of our cells, how does our immune system know that that's not a normal cell? That's a virus-infected cell. And so if you have a virus that got inside of one of your cells and it has these little blue circles all over it, those little blue circles that are on the inside will actually migrate to the outside. And so that our immune system can look at this cell and be like, those little blue circles are not supposed to be there. Those are viral little blue circles that are just being presented on the outside. So now our immune system will kill that cell. And then we do have what are known as autoantigen. Auto means self. So if you have an automatic car, most people do these days. I don't know if anyone has a stick shift anymore. Auto just means self. It shifts by itself. These are self-antigens. These are normal antigens. So these are things on the outside of our cells that in theory, they could trigger our immune system as foreign, but our immune system has recognized them as normal. So they're self-antigens. They're normal. They're supposed to be there, these little structures. Now, a couple more things about antigen. I can't remember if I moved the slide around, so if you look at your PowerPoint and you're like, where is this slide? It's later on. Because I used to talk about them later, then I'm like, nope, it's perfect time to talk about them when we talk about antigens. And these are antigens that are considered autoantigens. We give them their specific types of autoantigens, and we call them major histoability complex antigens, or we just shorten it to MHC. So these are glycoproteins. These are little sugar carbohydrate proteins on the outside of our cells, which again, in theory, the immune system could recognize it as foreign if it wanted to. And these little tiny protein things are on the outside of the cells of most cells of vertebrate animals that are not recognized as foreign. But what makes them different? Why do we call them MHC antigen, the major histoability complex? So every nucleated cell in your body, like each one of you, every nucleated cell in your body has an MHC, a major histoability complex, 1 antigen on it. But each one of yours is unique to you. Reason why we discovered these is when we started to do grafts and organ transplants, is that you have a very unique MHC1 antigen on every cell in your body, which means if I gave one of you an organ, because you needed an organ from another person in this room, your body is not going to recognize someone else's MHC1 antigen. And this is where we start getting graft rejections, why people that have different types of grafts or any type of organ transplant have to be given immune suppressants, because your immune system will always recognize someone else's tissue as foreign. That's the histoability complex. Histo means tissue. Tissues are not always compatible, because every single one of your nucleated cells has an MHC1 antigen on it that's unique to you. Now, I do say every nucleated cell in your body. There's only one cell in your body that doesn't have a nucleus. Any guess? I know, it did have a nucleus and then it lost it, which gives it its unique shape. Which is why red bloods look like they're caved in, is because they lost that big, huge nucleus. So they have that caved in structure. So every single cell in your body, except your red blood cells, has an MHC1 antigen. So like even your white blood cells, it doesn't matter. Every tissue cell, every white blood cell, every cell in your body has an MHC1 antigen that's identical for you. Now, MHC2 antigens. These are still antigens found on the surface of your cells, but not every cell. And this is why if you have this printed, you could draw a line. MHC class 2 antigens are only found on, that's my line, antigen-presenting cells. Body has class 1, nucleated. Only the antigen-presenting cells in your body have a class 2. These are going to become more important when we get into our cell-mediated branch on Thursday. But different types of cells that are considered antigen-presenting cells. Dendritic cells, and these are not the nervous tissue dendrite type things, but we have dendritic cells, a lot of them in our skin that are there to recognize foreign invaders trying to get in. Macrophages, this just means a big eating cell. B cells themselves can actually have the MHC2 antigens on them. And the idea is that these cells, whether they're dendritic, macrophages, or B cells, they will engulf whatever foreign invader is in there. So you got a piece of dust, you got strep, whatever it is. They will engulf that foreign invader, and then they will present the antigen on the outside of that cell to our immune system to be like, hey, I just ate this thing. Like, do you see it? Do you see it? Let me show you this thing I just ate you. Please get rid of it. Like, we need to get rid of this foreign thing that's in the body. And so they just present these little antigens to our immune system. Now we're going to really get into the humoral immune branch. So the humoral immunity branch, I'm just grabbing my clicker, is all about the B cells. Again, they arise and mature in the red bone marrow. That's where they get their B. Where we find a lot of these B cells, a lot of them in the spleen, a lot of them in the lymph nodes, and a lot of them in that mucosa-associated lymphoid tissue. A lot of that tissue in our digestive tract. Our blood does not have very many B cells. I mean, there are some there, they are patrolling, but most of the B cells are hanging out in the lymphatic vessels themselves. And the main job of a B cell is to make antibodies. Like, that's the ultimate goal of the B cells. This is my little refresher. Humoral immune branch uses B cells. The ultimate goal is to make antibodies. So what I'm going to hope to get through is get through the antibody slide, because there's more than one type of antibody, and there's a whole process to make antibodies. So the first is just looking at what does a B cell look like. So it's still a cell, still lymphocyte. It's got a big old nucleus. I'm just not going to draw it in there. And you have a lot of different B cells in your body. Structure, they all look identical to each other. You would not know the difference. I'm going to draw 3 of them. This is showing 3 B cells, and you've got like a million. I'm drawing 3. And each B cell makes one type of B cell receptor on its surface. So this B cell makes a B cell receptor that's red. B cell makes a B cell receptor on the outside that's orange. And they look like little Ys. And this third B cell makes little B cell receptors that are green. So each B cell makes only one type of B cell receptor. This one only makes red ones. This one only makes orange ones. This one only makes green ones. So each B cell only makes one type. But each B cell makes many copies. So one has like 5 on it. Someone's got 5, that one's got, I did 5. So each B cell makes many copies of that B cell receptor. It's a lot more than 5. Each B cell receptor only recognizes one antigen. This red B cell receptor, and I did color for a reason, this red B cell receptor can only recognize little red triangles. This one can only recognize little orange circles. This one can only recognize little green squares. So each B cell receptor can only recognize one thing. But ultimately, we can recognize millions of different things. We can recognize millions of different little 3D epitope structures. So that means you've got millions of different B cells in your body. Think of it like a million different colors, a million different shapes that these B cells can recognize. So many B cells, I compare them to snowflakes. Not every B cell looks alike. They're all slightly different from each other. It's because those receptors can all recognize something slightly different from each other. Each B cell is specific. You just got a lot of them. Zoomed in picture of what these B cell receptors look like, and yes, they do actually look like Ys. So this is part of our B cell. It's still a cell, still has a phospholipid membrane on it. And the B cell receptor, the whole Y has got a little anchor part. That little part of the Y, on the top part of the Y, I like to always, think of it's like the hands of my body is the Y. The hands are what actually recognize the foreign objects. So it's not the whole Y that recognizes the foreign objects, it's just the little tops of the Y that recognizes the foreign object and can attach to it. So this B cell receptor, big old Y on the outside of the B cell, the very top, the Y, the little hands, recognizes that triangle, that very specific epitope. And as soon as it recognizes it, again, it's like little hands, it'll attach to it. And now this B cell becomes activated. It's like, oh, I just finally encountered the thing that I'm supposed to attach to. I now become activated to do something. And it's the, what does it do once it's become activated? So the B cell becomes activated when the antigen binds to the surface receptors and cross-links them, which is really worded for the antigen. Whether that little triangle, that foreign object, cross-links, that just means binds to those little receptors. As soon as those little hands recognize the thing they're supposed to, whether they recognize, the little squares or circles or whatever, as soon as they bind to it, as soon as that hand binds to a foreign object it recognizes and it's supposed to, that whole cell becomes activated. So this is showing an example. They've got 3 different B cells. This one's got green, one's got, I think it's supposed to be dark blue or navy, and then a light blue. 3 different B cells, 3 different B cell receptors, and the red thing is the foreign object. Only one of the B cells can recognize that foreign object, and it happens to be the navy blue one. As soon as it binds to it, as soon as that hand binds to the foreign object, this is now an activated cell. And the first thing it wants to do is make more clones of itself. Again, you win a war a whole lot better if you've got a strong and very large army. So it starts to make more of itself. So now instead of having just one, now we've got 2. So you'd have a lot more. But now we have 2, and they're clones, so all the B cell receptors on the outside all have that exact same B cell receptor, that navy blue one. But they don't just stop there and just be like, oh, I'm an activated cell and I've got lots of me. The B cells will change, and they become what's known as an effector cell. Because an activated B cell doesn't in itself do anything. It's like, I got lots of them, but it doesn't do anything. It instead changes a little bit into different cells. And there's one of 2 different kinds of cells these activated B cells become. Most of them are going to become what are known as plasma cells. And the plasma cells don't have the B cell receptors on them. They are larger cells. But the job of a plasma cell is to make antibodies. It churns out tons of antibodies, 2,000 antibodies per second for every plasma cell. So again, you made lots of activated B cells. Most of them, in my picture, 3 out of 4, most of them become plasma cells, and each one, 2,000 antibodies a second for about 4 or 5 days. So that's a lot of antibodies. Then they die, but they did their job. They made lots and lots of antibodies. It's those antibodies, which we'll talk about in a little bit, they're the ones that actually circulate around in the body, and they're looking for the foreign invader. Now, the antibodies, and I'll make this picture bigger shortly, the antibodies that are made, how do they look in comparison to a B cell receptor? Antibody. Do they look alike? Yeah, they're identical to each other. They pretty much are identical to each other. Again, this B cell receptor, this navy blue B cell receptor, is specific, it's got its little hands on the end, to recognize this red structure, that foreign object that activated it. Well, if the job of this was to recognize that, the antibodies need to look the same too. so that they can also still recognize that foreign object. So those antibodies are trained, meaning they're structured identical to the B cell receptor, so they are now identical to recognize that foreign object and bind to it. Now, if an activated B cell didn't become a plasma cell and churn out lots of antibodies, which really is the ultimate goal of our humoral branch, the other thing they can become, memory cells. They still look identical to a B cell, but memory cells provide memory. It means these are B cells that are like, okay, I've seen that red thing before. I know how to make antibodies. So this whole process of recognizing a foreign object, activating the B cells, they become plasma cells, and churning out antibodies, the first time you're ever exposed to something, this process can take 2 weeks. Sick? 2 weeks? Man. Before your immune system is really at peak form, making tons of antibodies to get rid of that foreign object. But if you've got memory B cells, they're like, oh, I know what to do. We've done this before. As soon as I see this red thing, it activates the cells faster. They become plasma cells a whole lot faster, and you can churn out antibodies within 2 days instead of 2 weeks. And so a lot of times you can get antibodies out to fight the foreign invader faster than that foreign invader started to reproduce. The idea is, again, it fights it off before you ever even get sick. You didn't even know you had a foreign invader in your body because your immune system got rid of it so quickly. The bigger picture, I just made it bigger. So my little refresher before I get into some antibodies. Again, we have millions of different B cells. This picture shows 3. Millions of different B cells. All of them have their very own unique B cell receptor. Once the end of the B cell receptor recognizes its very specific thing that it's supposed to recognize, it becomes activated and makes lots of itself. Most of those activated B cells will become plasma cells, and they churn out tons of identical antibodies that can float around in your body looking for that foreign object. If they don't become a plasma cell, a few of them become memory cells so that we can recognize things and mount responses faster. Antibodies, the things that look like Ys, because yes, they are identical to the B cell receptor that eventually activated the whole process of making them. An antibody is a protein. It also has another name, an immunoglobulin. Same thing as antibody. Well, I know, because if you're like, what are immunoglobulins? Because the book will shorten them a lot to IG for immunoglobulin. It is an antibody. So if sometimes I write AB for antibody, sometimes I write IG. You'll see it, they mean the exact same thing. But antibodies are proteins. And so I know some of you guys are going into nursing. And so if you have a patient that's sick, they need protein in their diet. If you are churning out thousands of antibodies a second, if you've got a healthy immune system, those antibodies are all proteins. Those proteins have to come from somewhere. And the biggest source of it is the diet. So that's why you encourage patients to be like, if you're sick, you have to have protein in your diet for your immune system to really be at its peak form. They come from the plasma cells, the things that are churning out thousands of them a second. And yes, they're identical. They have that Y shape. Now, they have a variable region and a constant region. The idea is that the majority of it is the constant region. It's the hands, that top part, that's the variable. Because they all look really identical. It's just the hands that vary in what it can recognize. Do you have the hands that can recognize, little triangles, little circles, little squares, millions of different things. So that's where they get the variable part. But the body is pretty close to the same. Now their function. Again, we churn out thousands of antibodies every second for every plasma cell. And it's like, it's the antibody's job to get rid of those foreign invaders. But how? How do these little Ys go around and get rid of the foreign invader that got in? Well, they will bind to the antigen because they have hands that bind. And they will bind and recognize any bacteria, toxin, or extracellular virus. The antibody's job, I'll write more up here. B cells either become plasma cells or memory cells. And the plasma cells' job is to make the antibodies. B cells and antibodies are only recognizing extracellular when something's extracellular, it just means it's not inside the cell, it's outside of the cell. So these are things that are not hiding out inside of our cell. They can recognize bacteria, as long as the bacteria aren't hiding out in our cell, which most don't. They can recognize toxins that are floating around the body, and they can recognize any virus that's still extracellular. And so these antibodies are going to recognize any of these bacteria, toxins, or extracellular viruses and try to get rid of them. And how antibodies can get rid of them, a lot of them increase phagocytosis. Again, so then those things get eaten up and broken down on digestive. Or they can inactivate pathogens or toxins. And they can do it in multiple ways, which is my next slide. So the how they can inactivate pathogens or toxins is my next slide. So, one, as they bind, again, they're little Ys and they have their little hands when their hands bind, they can neutralize that foreign object. So they can work with what's known as neutralization. Just means that the antibody binds to the toxin or binds to the bacteria or virus on some critical portion. And now that thing can't cause the disease. It can't bind to other things. It can't create, whatever toxic effect that toxin had. It just neutralizes that bacteria, the virus, or the toxin so that it can't cause any harm. And eventually our immune system, we'll get into it, we'll get rid of it. Another is called opsonization, and this is the phagocytosis. Everything's like Latin and Greek. Opson means to eat, or phago. So opsonization is going to be anything that has to do with phagocytosis. Again, some of these organisms, whether it's a bacteria, a virus, or even a toxin, sometimes they can be small, sometimes they can be slippery, and phagocytes, the eating cells, can't always digest them or get around them very well. They're a little slippery, they're not very big. However, if you're a bacteria or a virus, extracellular, or some type of toxin, and you were kind of small or kind of slippery, but now you are covered in little antibodies, you're not very small and you're not very slippery anymore. And so it increases the chance for a phagocyte to eat it. So just by covering things in antibodies, increases the chance they're going to get eaten. They can also do agglutination. So these antibodies, because they have two hands, that doesn't mean they have to, both hands have to attach to the same bacteria. You could have one antibody that attaches to two different bacteria, and then another antibody attaches that bacteria to another one. And all of a sudden you get a big grouping of bacteria. Agglutination just means gluing or clumping together. Well, by having big clumps of bacteria, one, it can increase the chance of them getting recognized by phagocytes, but it will also more likely, these things will get filtered out in the spleen as they travel through the lymphatic system. It's another reason when you get sick, your spleen enlarges, not just lymph nodes. And so they get filtered out and then attacked in the spleen a whole lot easier by clumping them together. Another way they can work, there's lots of ways these antibodies can work, they're all so clever. One, it's called antibody-dependent cell-mediated cytotoxicity. So big. It just means the antibody is going to do something, and it's going to target the cell-mediated branch, which we'll get to Thursday. And it's going to cause some of those T cells to kill the bacteria, the toxic. So if an antibody attaches to this bacteria, there are some antibodies on the body part, that constant region, that normally doesn't do a lot, that constant region can actually attach to another type of cell. It's called a natural killer. That's where the NK stems from. A natural killer lymphocyte. And guess what the job of a natural killer lymphocyte is? Based on its name. Just kill bacteria or some foreign object. So if an antibody is attached to that foreign object and, oh, well, it's not getting eaten and it's not getting filtered, one of the natural killer cells can attach to the body part of the antibody, and the natural killer's job is to secrete, let me say, not a chemical that causes holes in the bacteria. And it's never good if you're a bacteria to have holes in your outer membrane, in your cell wall, and so ultimately the bacteria will die. And so the natural killer cell will kill the bacteria. So it's like the antibody just recruits some help of a natural killer cell. The other one didn't have a picture. Antibodies can also activate complement and inflammation, which is part of our second line of defense. So by having antibodies being produced and attaching to foreign objects, just by having them produced and attaching to foreign objects, the antibodies activate complement proteins, second line of defense, and they can increase inflammation. Again, it's, well, our third line of defense, the humoral branch isn't doing anything, so let's recruit some help from our other areas of our immune system. Inflammation is there for a reason. Fever is there for a reason. It's good things. It's there for a reason. Complement proteins also increase the chances of phagocytosis happening and increase the chances of fever happening. Again, everyone's like, fevers are bad. Are they though? They do decrease the chance of, bacteria reproducing. It's good, as long as it's not too high. I think I don't know if I gave you guys this slide, if you wanted to write notes down. But there are 5 classes of antibodies. They all look like Ys, but sometimes they hang out in different areas and they're kind of shaped and prone based on where they are in the body to do different things. So immunoglobulin, again, same thing as antibody, G, and they're all named for Greek letters, so the immunoglobulin G is gamma. It is the most common of all antibodies. So if we took a blood sample, you would see some Ig, G in your body. It's the longest lasting. Again, antibodies, once they're made, their job is to target the thing, get rid of it, and then they will eventually start to go away. But the IgGs last the longest. We find it in the blood serum, the plasma, and the IgG can cross the placenta and give even a developing fetus some immune system support. IgM, which I think it looks like the coolest one because IgM antibodies always hang out in groups of 5. So they're called pentamers, and they look like a big old snowflake. So you've got 5 antibodies. Each has 2 little hands that can attach to things. So ultimately, these things can attach to 10 different objects. I mean, the same kind of object, because they all recognize the same thing, but they can attach to 10 bacteria if they need to. It's also the first antibody produced. So if you have been exposed to something, the very first one that will show up in your immune system is going to be the IgMs. So it's also called the first responder. Because of its shape, because it can attach to 10 bacteria all at the same time, its top job to getting rid of that organism is agglutination. I mean, it can do a ton of clumping. Imagine if you had 10 hands, you can form these big, huge clumps of bacteria for getting filtered. The IgA, it's the alpha in the Greek letter. You don't need to know what they stand for. The IgA, its kind of claim to fame is IgA is found in all body secretions. Your sweat has antibodies in it, specifically antibody A. I'm like, any fluid that our body can make has this particular antibody in it. I'm making only antibody A. So all the other secretions that you make are not going to have all the antibodies, but they will all have antibody A. So it's associated with body secretions. So your saliva, tears, breast milk, colostrum, semen, vaginal secretions, any secretions for your digestive tract. I mean, any secretion that our body can make, sweat, it's not listed up there, makes antibody A. This allows us to have lots of protection everywhere in our body. Through any kind of secretion, we're releasing antibody. But it is this particular antibody, when they say, breast milk is best, it's because if mom was exposed to something, they had antibodies against it, and is breastfeeding, that baby is also now receiving that same antibody and can also fight off whatever mom just had to fight off. And so babies don't have a very well-developed immune system, and they don't have a lot of memory cells because they haven't been exposed to anything yet. And so since they don't have a great working immune system right away, by having antibodies that mom made, they do actually get a lot of protection that way. The antibody E, for epsilon, don't care, it's involved in allergy responses. It's what triggers a lot of your histamine, like overproducing of histamine when you have any kind of allergy response, which is why if you're having an allergic response to something, you take an antihistamine. It's because this particular antibody is triggering your basophils from making lots of histamine. It's also increased and active at attacking parasites, specifically parasitic worms. Now, in your second line of defense, if you haven't gotten to that video yet, one of our white blood cells is also increased with allergy responses and parasitic infections. Allergies and parasites, like parasitic worms, it's like two completely different things, but the cells that work against those are the same. I don't know why. Your eosinophils, starts with an E, also are responsible for allergic responses and are activated when you have a parasitic worm infection. And then your antibody E is also, they both have an E in them. That's how I tie them together. Yeah, and they're totally different responses in how they work, but allergies and parasitic worms. IgD for delta is an antibody that's actually found on a B cell. They don't exactly know its full function yet. They think it's still for attacking, it's still for increasing phagocytosis, still for increasing neutralization. But the interesting part, because this is an antibody and it's found on the surface of a B cell, how do you know that this is an antibody and not a B cell receptor? Because they look identical to each other. Again, it's still there to attach to things, to recognize things, and increase getting rid of it in the body. We don't know everything. We just don't know everything what it does yet. They're working on it. That's where I'm ending today, because that's enough. Let me stop the recording.